thank you everybody for joining today. I'm absolutely delighted to be able to welcome you to our session to discuss what we do in the Centre for Inflammation Research and Translational Medicine, or CIRTM as it's affectionately known. Our centre's been running now for just over a year, and I guess this is, for one reason or another, the first main opportunity, I would say, really to present what we do in the centre. So we're very excited to be able to do that. We've got we've popped the link, the web link to the centre information, and I ask you to go away and take a look at that. It's got a lot more detail than what we can present in this morning's session. So you'll be able to see a little bit more about the centre members, what we do, some current projects and some projects that have now passed, but it gives you a real flavour for what research goes on within the centre. And feel free to tweet us as well. Okay, so our mission really is to promote interdisciplinary research into the understanding of the inflammatory and immune processes that really underpin human health, disease and healing, and really to provide an outstanding environment for research training in the field, and also to translate and take forward potential therapeutic strategies for the treatment of a number of different types of diseases. And we do this in a multidisciplinary approach. We look at sort of preventative and therapeutic strategies that really cross over the fields of inflammation, of disease, and this sort of perturbed immune system that occurs in a lot of disease states, and we'll talk about that throughout this morning's session. And also that perturbed immune system follows with aging as well. So it's also a physiological setting where some of these issues occur. So who are the members of the centre? Currently we have 13 members. We've got one additional one that will be joining us in September, I'm pleased to say. I don't need to say everybody's name, you can see them there on the screen, but as I said before, it's please do go to the website and have a look at the individuals and their research areas. Those names that are highlighted in red are going to present some of their research to you today. So you get a bit of a flavour of what the diverse range of the research that's going on within CIRTM. So we really do tackle global problems within the centre. And so there's different areas that we all work on, and some of them are collective areas, and some of them are individual areas. So we focus on cardiovascular diseases, obesity, cancer, mental health disorders, TB and other infectious diseases, antimicrobial resistance and drug safety. And that concoction and collection of different disease states are really global problems, and that's the reason why we within the centre are trying to tackle these major issues that affect not only the UK but also worldwide. So I hate to throw stats at you, but I feel that sometimes it puts things into perspective, and I think that's very important because we're doing the research for very specific reasons. We're not just doing it for the sake of doing it, but because we want to help society, because that is our end goal, to be able to understand disease, to understand physiology as well as the pathophysiology of disease, but also obtain therapeutic strategies to treat patients and to help society in general. So we put together a few stats, and I have to say that I thank the team that have put these statistics together. So thank you for that. But you can see that, I'm not going to go through all of them, but you can see that, for example, in cardiovascular diseases, 450 deaths every day from heart and circulatory disease. Somebody has a stroke every 5 minutes, and in fact, every 40 seconds, somebody dies of a stroke. So these statistics really show the problems that we're facing. With obesity, 21% of children at the age of 10 and 11 are obese, and 15% of all paediatric cancer deaths are accounted for by neuroblastoma. So these are really quite very troubling, not quite troubling, very troubling problems that we face. Mental health disorders, and that's all in the press at the moment, in particular with the COVID situation that we're all facing. 1 in 4 people experience mental health issues each year, and despite the fact that we are better equipped at not necessarily dealing with, although we're getting better, but talking about mental health issues and the importance of those that affect society and individuals throughout their livelihoods. You know, those statistics are worrying. The World Health Organization has declared that antimicrobial resistance is one of the top 10 global public human health threats, facing humanity. If you look at TB in the world, 1.4 million people have died from TB in 2019. And then if we focus on COVID, you know, that's at the forefront of everyday events, that we've got 3.23 million deaths that have been associated with COVID since 2020. So what you can see with just showing some of those statistics are really the global health, not just UK, but global health problems that we're facing, that we're trying to tackle and challenge within the centre. And through today's presentations, you'll hear a little bit more about how members of the centre are trying to do that. So how do we do what we do? We use a range of techniques and methodologies. We look at from bacteria to worms to zebrafish, mice and other species, and then humans. So we take a sort of bench to bedside and back again approach, which is very important to understand the translational aspects of everything. The slides here really not just to say about the models that we use, but also some of those models, such as in vivo models, animal models, and humans, bring with them ethical challenges, and that sort of information that they bring is very important, and we want to be very transparent about the types of models that we use within the centre. We do everything when it comes to both the in vivo, so working with animals as well as with the humans. It is all under very tightly regulated ethical governance. For the in vivo work, it's Home Office driven. And we work very closely with the National Centre for the Replacement, Refinement and Reduction of Animals in Research. That's the NC3Rs. And we do that because before we go into an animal, before we go into patients, we have tested compounds and those strategies before we get there. You know, so everything that we do for a very specific, very controlled, very regulated reason. And we're more than happy to talk about the ethical concerns and considerations in those models that we use. Happy to do that. So we take a very integrated understanding of health and disease within the centre. We go from genes to the molecular, to cellular, to organ networks, and finally population networks. Using multidisciplinary approaches, we've listed some of them there. We've got multiomics, we've got big data analysis and bioinformatics, all the way to sort of imaging, novel imaging modalities, such as intravital microscopy. So lots of different techniques in order to really answer and try to help with those global challenges that I presented. So as far as potential interdisciplinary research and some ideas to perhaps whet the appetite, we've come up with a possible list of things that where the centre could fit with other areas within the college and beyond. We've got computer sciences, so we do a lot of big data analysis and obviously AI that comes with that. With engineering, microfluidics, we'll talk about organs on a chip. I think Ashley will talk about that. So microfluidics, looking at cellular trafficking, looking at using in vitro models to be able to look at different aspects of that. Social sciences, we've got the social dynamics of inflammation and behaviour. I think Luigi will talk about depression as well as inflammation. Medical sciences, we've also got that translational and clinical studies. You know, what we do in the centre, the disease models that we use translate to the clinic, and so the interaction with the medical school and our clinical fellows is very important in helping us to achieve our objectives. So that's it from me with respect to the welcome and introduction. This is today's agenda that's there. So what we've done is we've divided it into sort of 4 sections. And so we're going to have some talks from members of the centres. They're going to talk about their research. And the first selection is going to be on infection and immunity. So Ronan, Steve and Ashley will present in that. And then we'll move forward into the cancer fields, and Arturo will present on cancer and immune dynamics. And that will be followed by Luigi, who will look at drug safety and immunomodulation. And finally, we'll focus on cardiovascular diseases, and Raha and myself will present research within those fields that we're doing in our labs. We thought what would be best was, rather than take questions at the end of each member's presentation, we'd do a collection at the end of the actual sort of panel discussion. So please feel free to pop questions in the chat and we'll get to them at the end. If we're running over time and we don't have enough time to answer all the questions, we will do so. We will get back to you so that you'll have answers to your questions. And in the meantime, sit back and enjoy, and I hope you find it interesting. I hope it whets the appetite. I hope we get some active discussions and hopefully some networking and possible collaborations out of this morning's presentations. So without further ado, I shall pass you on to Dr. McCarthy, who's going to talk about antimicrobial resistance out of the first of 3 talks on infection and immunity.
So I'm going to talk a little bit today about antibiotic resistance and the problem or the threat that poses to our global healthcare systems. So back in the middle 2000s, the Cameron government initially commissioned a report to really take a hard look at antibiotic resistance, because clinical professionals were saying this is going to be a serious problem facing humanity over the coming decades. And the report that was produced was called the O'Neill Report, and it made for some very startling reading, because it really interrogated the threat that antibiotic resistance poses with respect to human health. And it demonstrated that by 2050, if we don't do more to tackle antibiotic resistance and antibiotic resistant infections, it's estimated that we'll have 10 million deaths per year due to antibiotic resistance. So that's more than cancer, diabetes, measles, tetanus, all these other what are considered high impact conditions in terms of human health. So the threat that antibiotic resistance poses is really twofold, and it really comes from a number of different areas with respect to every time anybody goes to a hospital. If they have a wound, or if they're having surgery, or if they're having a catheter, all of those are potential opportunities for infection to occur. And the wider medical field is now beginning to understand the real threat that infections, let's say for cancer patients or for even dentists doing a tooth extraction, if they get an antibiotic resistant infection, that's extremely difficult to treat. And a recent poll of oncologists showed that 95% of them are worried about the impact that drug resistant bacteria will have on their future cancer treatments and the capacity to care for their patients. So while there's kind of initially an insular view that antibiotic resistance just impacts people who get wound infections, it also has ramifications for wider medicine in general. So why do we have antibiotic resistance, and what's happening? Well, we had a scenario in the 50s, 60s, 70s, and 80s where antibiotics were routinely being discovered. And there was kind of a relaxed attitude about antibiotic resistance, because while it was emerging in the clinic, we were always finding new antibiotics. Researchers were finding new antibiotics, which meant that we could combat the resistance with a new antibiotic. However, what started to happen in the 80s and all the way up to the current time is that we couldn't actually find new classes of antibiotics. So essentially we discovered all the, well, not all, but we discovered the majority of the freely identifiable antibiotics. So this is creating a problem now where bacteria have continued to evolve, and in the clinic they're continuing to see bacteria that are resistant to antibiotics. And there's cases that have been identified over the last couple of years of patients who presented with an infection that's resistant to every single antibiotic that we have on the planet. And there's very little that you can offer a patient in that scenario beyond palliative care. And that's quite a scary scenario that really is, I suppose, coming into focus with respect to the next couple of decades, a post-antibiotic era, an era where we no longer have antibiotics that can be given to patients, where antibiotics can no longer be used prophylactically. And if that scenario does occur, we go back to a situation that we had in the 1900s, where you could very easily die from a simple cut that you get shaving or from a tooth extraction. Because if you get an infection and we've no drugs to treat it, then essentially there isn't much options available to the patient. So how are we tackling this in my lab? Well, in my lab, we focus in on two specific pathogens. We focus in on Acinetobacter baumannii and Pseudomonas aeruginosa. Now, these two particular pathogens are extremely important because in 2018, the World Health Organization published a list of priority pathogens, and it basically ranked the biggest threats to human health with respect to antibiotic-resistant pathogens. And the top two pathogens that they identified as priority one critical pathogens were Acinetobacter baumannii and Pseudomonas aeruginosa. Now, both of these bacteria are capable of causing infections, primarily in opportunistic infections. So they're very frequently isolated in hospitals, infecting the most vulnerable among our population. So one of the key things that we look at in my lab is how these bacteria resist antibiotics. And one key mechanism that both of or almost all priority pathogens have in common is the capacity to form a biofilm. Now, a biofilm is like a community of bacterial cells. And just like humans, when we live in a community, we're much stronger, we can overcome challenges much easier. It's the same for bacteria. So when they live in a community, they can overcome challenges such as changing environment, exposure to biocides or disinfectants, and critically antibiotics. So how a biofilm forms is that a bacteria in step one here will attach to a surface or to each other, and then they'll start to produce something called an exopolysaccharide matrix. And that matrix can be thought of simply as a force field. It protects the bacteria, encases the bacteria, and then subsequently, if you treat the bacteria with antibiotics, many of the antibiotics either get caught or are repelled within this exopolysaccharide matrix or within this force field. And evidence has suggested that bacteria growing in these communities are between 10 to 1,000 times more resistant to antibiotics, disinfectants, and our immune systems. So this presents a huge challenge with respect to treating bacteria, but it's also an opportunity because if we can stop bacteria forming a biofilm, we can very quickly make those bacteria much more treatable. And that's something we focus in on quite a bit within my research group. Now, many of you have probably heard of biofilms. The plaque that you routinely get on your teeth is an example of bacterial biofilm formation. But within medicine, they're found throughout the body, particularly in implants, such as hip implants, catheters, pacemakers. And a key area that we focus in my lab and that I'll go into a little bit more detail on today is actually wound-associated biofilms. So up here in the top right-hand corner, you can see a burn wound that has a biofilm formed on it. We're very interested in trying to explore how can we stop these biofilms forming, and if we stop these biofilms forming, do we make the treatment or the amount of time that that person has to spend in hospital shorter, and do we improve their survival prognosis? And in fact, 80% of hospital-associated infections involve some element of biofilm formation. So it's extremely important that we develop new strategies to stop this mechanism of overcoming antibiotic resistance or antibiotic treatment. Now, to study biofilm formation, there's a range of different model organisms are used. The classical lab mouse model, the rabbit model, and in some instances, the porcine model are used. But within my research group, we have a strong emphasis on trying to move away from these ethically challenging models to models that are more ethically friendly. And we've done this in this instance, whereby we use an alternative model called a wax moth or Galeria melanella. Now, this model is free from the ethical restrictions. It's not covered by Home Office restrictions. And this allows us to do high throughput screening strategies to look for new drugs, which you can't possibly do in some of the other animal models. And also the models that we're trying to develop using this invertebrate will hopefully reduce the numbers of mice and other model organisms that need to be used in these studies. So what we wanted to do was establish if we could use Galeria melanella or the wax moth to study burn wound infection. Now, this particular model organism has been well used and well characterized as an excellent model to study a wide range of different human pathogens, but it's never been used to study burn wound pathogens. So what we did in my lab, and we had a very talented student who developed this assay, we actually burned the backs of the Galeria and we added bacteria to the actual burn wound site itself. So this was replicating what would happen if a human got a burn and that the bacteria encountered the burn wound site and caused an infection. And what we saw with this was that as the bacteria started to colonize the burn, the larvae started to get sicker and sicker, and the change in color or the melanization of the galeria represents how sick they've become. And ultimately, you can see that the infection progresses to a point where the galeria actually succumb to the infection. So we were really, really excited by this because this demonstrated for the first time that the galeria or the wax moth model could be used to study burn wound infections. But we wanted to take this a little further. We wanted to understand how similar is this insect burn model to human burn wound infection. So to test this, we tested some classical paradigms that are associated with human burns and animal burns. And the first of those paradigms, which we present over here on the left, is that the bigger the burn, the less the chance of survival. And we tested this by increasing the size of the burn that the larvae had and monitoring how they survived over time. And on the green chart line here, you can quite clearly see if they have one burn, they survive reasonably well. If we double the size of that burn, their survival starts to decrease quite significantly. And if we triple the size of the burn, we can see that they no longer survive past 55 hours. What we also wanted to test was one of the key prerequisites for treating a burn wound patient in the clinic is fluid resuscitation. They need to receive fluid resuscitation as quickly as possible after the burn has occurred. And what we demonstrated using our model is that if we give fluid resuscitation to the insects, they actually survive the burn procedure much, much better. So we're very excited by this because it demonstrated that the insect burn wound model has a number of different similarities that make it a good model to study human burn wounds. So we then wanted to see some of the classical pathogens that are very prevalent in human burn wound infections. So we tested Pseudomonas aeruginosa, one of the most common burn wound infections, and we saw that if we applied Pseudomonas aeruginosa to the burn wound itself, we could see that it colonized the burn wound and that the larvae no longer survived. We also tested Mrsa, as many of you are well aware, one of the key antibiotic resistant pathogens that's commonly found in hospitals. And we also saw that applying that to the burn wound caused a significant decrease in survival. We also tested some skin commensals, so bacteria that are commonly found on the skin. And in this instance, we saw Staphylococcus epidermidis, and we saw if we applied this to the burn wound, we didn't get a significant decrease in survival. So how are we going to use this model to develop new treatments for burn wound pathogens? Well, one aspect of my research is looking at the skin microbiome. So over the last 20 years, we've become well established that the gut microbiome plays a key role in human health, and that we have a huge number, up to 1.5 kg of bacteria living in our guts, and they play a key role in preventing infection and also allowing us to get nutrients from our food. But another microbiome that we have is the microbiome that covers our skin. And we have a layer of bacteria and microorganisms that covers our skin. And that barrier is a protective barrier that protects us from potential other skin pathogens. Now, if somebody gets a burn wound, that key barrier is disrupted, obviously, and there's an opportunity for pathogens to colonize. So what we wanted to test was, could we take some of the good bacteria that we have living on our skin and apply them to a burn wound and hopefully prevent infection? I'll just show you a snapshot of some of that data that we have demonstrating that it is actually possible to do this. So on the blue line here in this graph, this is larvae that were infected with Pseudomonas aeruginosa, the common pathogen, and as you can see, they start to die quite quickly after being infected. In the yellow or in the green line up here, we have our skin commensal, our good bacteria that we've applied to the wound, and you can see it doesn't really kill any of the larvae. The red line is a critical one. The red line is larvae who were burned, and after they've received the burn wound, we very quickly applied the commensal bacteria to the burn wound and then tried to infect the larvae with Pseudomonas. And what we saw, which was we find this quite remarkable, is that applying this good bacteria to the burn wound immediately after the burn wound has happened, we can increase survival and prevent infection by up to 60%. So we're hoping that this may pave the way for skin probiotics or bacteria that could be potentially applied to burn wounds to prevent infection, limiting the use of antibiotics in the future. So this is just a snapshot of some of the research from my lab. We've been supported by a range of different funding organizations, and we have a range of different international collaborations that we're working on this with. So I'd be delighted if anybody has any questions to put them in the chat box, and I can address them at the final stage. So I'd like to pass on now to Dr. Smith, who will talk to you about some of his research.
What I'm going to try and do today is give you a sort of a taster of the type of research that I am interested in with respect to tuberculosis. So as Felicity very nicely introduced, the centre has a strong focus on infectious disease, and particularly tuberculosis. So you can see here, this is a very nice image of the bacterium itself. So TB is caused by a type of bacterium of the genus known as Mycobacterium. And one of the particular characteristics of that bacteria is that it's got what we know, or what we call, a very waxy cell wall, a very waxy coat. It's very rich in lipids and fats, and that means it's very persistent. It's been with us for millennia as a human infection. It's particularly good at avoiding immune responses and our attempts to rid our bodies of it, which makes it a very effective pathogen. And so that is what leads to the challenges in terms of developing vaccination against it, and why it is still with us after all these years. This will be a good point to also mention that as well as the work that I do in CIRTM, we have other CIRTM investigators working on tuberculosis. So I would highly recommend that you go to the website and look up the work of Dr. Anthony Talaki. So his interest is in, and if I get this right, I don't want to misrepresent, but he works on more the molecular biology of tuberculosis and its pathogenesis. And we also have Dr. Ansar Pathan, who, like myself, works on vaccination for tuberculosis, and he's worked on some of the clinical trials of some of the novel TB vaccines. But as you will see, I am more interested in the oldest vaccination that we still currently have, which is against TB, and it's known as BCG. It's always worth reminding ourselves of some of the TB figures, facts and figures. Felicity already mentioned some of these in the introduction. So the WHO releases a global TB report every year. These figures are from the last but one report, the 2019 report. But the 2020 report, which recently came out, shows that these figures haven't really changed very much. And they make for quite sobering reading. You know, even in the times that we're living in, TB is still a top killer worldwide of any infectious disease. It kills around about 1.5 million people a year. I think Felicity said the more recent figure is 1.4 million, so it's coming down, but very slowly. And a lot of those deaths are accounted for by people who are co-infected with HIV. So HIV affects our immune response to TB and makes people much more susceptible to it. But possibly even more sobering is the fact that up to 10 million people every year fall ill with TB. So getting infected with TB isn't enough necessarily to cause people to fall ill. A lot of people are latently infected with TB, but these 10 million people are all people who are infected and contract active TB disease, which makes them very sick. And so what we need are three things, basically. We need better diagnostics against TB. We need better antibiotics, as Ronan's just very nicely portrayed. We have a serious antimicrobial resistance problem, and TB is not immune to that. We are not immune to TB. There's a lot of antimicrobial resistant TB in the world. And we need better vaccines. However, just before I go into the vaccines, it's worth considering the impact of COVID on TB. So clearly the world is very much focused on COVID at the moment, but there is a great fear that, you know, when we get past the pandemic, there's going to be a severe effect on our ability or on our progress that we've made to control TB. And so this graph, which is from the recent WHO report, shows what could happen if COVID affects our ability to detect TB cases. So you can see that even a relatively modest effect on our ability to detect TB, so a decrease in detection rate of 20 to 30%, if that persists for more than a few months, as is probably going to happen given the length of time that the pandemic's been with us, you can see on the right hand side there the effect of that on excess TB deaths, and it quickly mounts up. If you bear in mind that we currently already have 1.5 million deaths a year, and very quickly these numbers, that figure will start to increase. So this is something that is going to be with us and something that we have to cope with, and just brings us up short when we consider that we have been making progress on TB in bringing down deaths and infection rates. So I mentioned vaccination. We have a vaccine against TB and it's called Bcg. So this is a big year for Bcg. It's its centenary. It's the 100 years since it was first given to humans in 1921. It was developed by these two chaps, Carl Met and Guerin, who cultured the agent that causes TB in cattle, Mycobacterium bovis. They cultured it for long enough that it became attenuated, decreased pathogenicity in humans. We were able to administer it to people, and it induced a potent immune response that was also active against TB itself. And so this timeline just gives an indication of how TB use has developed over the years in terms of the clinical trials that were first held, the big trial in the UK in the 1950s to the 1970s. And through the time that Bcg was incorporated into the EPI, so the global infant vaccination regime. And it is now currently given in this country to infants that are at risk. So one of the issues with TB and Bcg particularly is its efficacy. So the fact that it's been with us for 100 years suggests that it does do some things well, and it can indeed protect us from TB. So the issue is that it doesn't always work as well. It depends where in the world it's administered and to what age group, and to other factors that can vary in terms of our exposure to different infections in the environment. All those things can affect how well Bcg might protect us. And so this figure, which was from a review a few years ago, just highlights exactly what can cause variation in Bcg efficacy. So the closer these bars are to this line, the further to the right, the less effective Bcg is. So you can see here that one of the things that affects it is latitude. So the further away from the equator Bcg is given, the better it seems to work. It seems to work very well in neonates. It works well in school-aged children, but only if they've been stringently tested for any previous exposure to mycobacteria. So the implication of this is if people have been previously exposed to other mycobacteria, then Bcg doesn't work very well. And so those are the main things that affect it. And so the challenge is to develop either an improved format for Bcg or a completely new vaccination approach that will circumvent these limitations for Bcg. And so what I want to do just in, I think, the last two slides is just give a little taster of the type of work that I've been doing on Bcg. So one of the advantages of this situation, whereby there are settings in the world where Bcg works very well and other settings where it doesn't, is that we can do comparative immunology in these places. And we can use this differential to teach us about what it is that Bcg does well, how it can induce positive, effective responses from our immune system, and what it fails to do in the places where it doesn't work. And so I've done a lot of work in my previous posts. I've only been at Brunel since January last year, so this is the work that I'm now setting up in my new lab at Brunel, is comparative immunology studies with collaborators in countries that are primarily closer to the equator. So this is a tweet that was put out. So this is partly to promote the CIRTM Twitter handle. So we tweeted this paper that was published relatively recently, and it describes a study that we carried out where we tested the immune response in infants from Uganda and compared those to the immune responses in infants from the UK, and what we're able to do. So I'm primarily focused on human immunology, and so it is possible to take blood samples and deduce a lot of useful information from the immune cells that are in those blood samples. And so in these studies, so this is one study, and there are previous studies in Uganda as well as in Malawi, another equatorial African country, where we've shown differences in the types of signaling molecules that immune cells secrete that indicate that the immune responses may be less effective, or why they may be less effective in these places. The second slide I wanted to show is to give a little bit more detail about the type of immunology that I do. So I know that this is not an immunology audience, but I do feel a little bit on safer ground nowadays talking about immunology than I did maybe a year ago. I think we've all become sort of immunologists to some extent in the previous year. And so I feel that I can talk a little bit about the different types of immune response. So you may know, you may have been following the news and absorbed some immunology knowledge and realized that it's what we call the adaptive immune response that gives vaccination its potency. So it's the adaptive response that allows our immune response to remember past infections, to remember previous vaccinations, and then enables us to combat infections when they occur. However, a more recent discovery in the immune system is the fact that the other arm of our immune response, what we know as the innate immune response, can also be a target for vaccination. So this, just to explain this slide, this is just a paper I found very recently, and it highlights two things. It highlights partly this aspect of BCG, whereby it can target the innate immune response. So in this title, the term pathogen agnostic effects, what that's suggesting is that BCG can induce immune responses that are not only effective against TB, so it's target disease, but also other infectious diseases as well. And neutrophils are one type of innate immune cell. They're not actually the type of innate immune cell I've mostly been interested in. So most of my work so far has been looking at what cells are known as monocytes. And so this leads to the second point I wanted to make with this slide. In CIRTM, I think, and this highlights that we're particularly well placed to start addressing some of these more novel ideas in immunology and vaccine immunology. And you will hear from Felicity later her strong interest in neutrophils, and I feel like this is a sort of a very advantageous situation because we can bring together our respective expertises to sort of start looking into these new discoveries in particularly in BCG immunology, but for vaccination immunology in general. Okay, and that sort of, as I say, it was just a snapshot, but I hope that gave you a good flavour of what it is that I'm interested in and the type of work I do. So at that point, I think, as Ronan said, if you pop any questions in the chat, we can address those at the end. And I will pass on, I think, to Ashley.
So yeah, thank you everyone. Thank you, Stephen and Ronan previously. So I'm going to talk to you today about bacterial vaginosis. This is quite a poorly understood and discussed disease. It's a disease that not a lot of people will be openly talking about due to the nature of it. So what is bacterial vaginosis? It's actually the most common vaginal infection that affects women between the ages of 14 and 49. And typically about 10% of women, but up to greater than 50% of women will suffer from bacterial vaginosis. And this is really dependent on the country they live. So the developing world, the incidence is much less. In the developing world, sorry, there's a higher incidence of bacterial vaginosis. And there is some evidence that certain ethnic groups may suffer more from this, but data is a little bit sketchy on this at the moment. And what bacterial vaginosis is, the vagina is normally dominated by Lactobacillus species. They make up about 95% of the bacteria in your vagina. You may have heard of them, like friendly bacteria that you get in your gut. They line the vagina and protect you from infection. When you get bacterial vaginosis, this is changed with by other bacteria, such as anaerobic bacteria, such as Cardinal, as you can see in the figure at the bottom. And they change what's going on, and it can result in you getting the fish odour associated with bacterial vaginosis, you get vaginal discharge, you get discomfort, amongst other things. And as you can see here, the normal vaginal epithelial cells on the left, and when you've got bacterial vaginosis, you get a lot of bacteria sticking to the cells, you get these cells coming off the vagina, and this is basically bacterial vaginosis. With bacterial vaginosis, apart from the discomfort and sort of embarrassment of the disease that people have, there are other more severe complications. It can cause pelvic inflammatory disease. People who have bacterial vaginosis have problems with pregnancy. It can cause infertility. It can cause people to have premature births and miscarry if they've got bacterial vaginosis. Also, people who have bacterial vaginosis are more susceptible to other STI infections. So if you've got bacterial vaginosis, the chances of picking up HIV is increased massively, and things like this. So people may know, may not know, that this is normally treated with antibiotics, creams, probiotics, things like this, which you've heard of, to try and sort of help with the symptoms. However, 60% of people who have bacterial vaginosis will get it again within 12 months of clearing it up. So this basically means we have no real effective treatment for this condition. We can alleviate the symptoms a bit, get it to go away for a while, but we can't cure. What's going on? This is the real crux, is what is going on. This is a really common disease. It affects a large proportion of women. It keeps on reoccurring, and we don't have an effective treatment for it. People have researched this for over 60 years, and they identified that the bacteria Gardnerella vaginalis was as the pathogen responsible. But recently, more researchers found that this is actually controversial, and this may explain some of the problems we have with treating bacterial vaginosis, and I'll go into this a bit more. I'd say thanks to Dr. McCarthy, Ronan, who talked about the microbiome, and so you hopefully know a little bit about microbiome already. He said the skin, and we talked about the gut, but basically we have different microbiomes in different parts of our body. We're oral cavity, things like this. And when you get a change in your microbiome, this results in disease. So for example, here you can see this top pie chart is the microbiome of the stomach. The different colours represent different bacterial species. When you have a stomach ulcer, which you may or not know about, is caused by Helicobacter pylori, you get a massive change in the bacteria that are present there. And you can see here for this pie chart, it's all gone green. This is the infection, it's changed the microbiome, you're unwell. If you look at the vagina, it has its own unique microbiome. I've talked about it's 95% lactobacillus. But when you have bacterial vaginosis, this microbiome changes. And we're not really sure, now we've done this microbiome research and found out all the bacteria present, it's ambiguous. There are over 400 different species of bacteria present in the vagina. Although yes, Gardnerella, when you have bacterial vaginosis, is the first to create a biofilm. Ronan talked about the creation of biofilms and how it protects bacteria from being cleared. But if you look, Gardnerella is present in 80% of women who don't have bacterial vaginosis. So this bacteria can't be something that's coming in and causing bacterial vaginosis. It's already there. So we basically need a new novel platform to be able to study bacterial vaginosis. Currently, we use two-dimensional models. This is where we grow tissue culture, so we grow cells in a plate, but they're not really truly representative of what happens in vivo. We've talked about animal models and trying to replace animal models. For the vagina, animal models are not very good. For example, mice, everyone uses mice in animal research. The mouse vagina doesn't have lactobacillus. So how can we study something that replaced lactobacillus in a mouse model if it's not there in the first place? So there's basically no realistic model to study pathogens that affect the vagina. So Felicity mentioned, I'm talking about our organ on a chip thing. So vagina on a chip. What we're doing is we're trying to create a model system which we can use to study bacterial vaginosis. And to get this, we need to grow cells. We want them in 3 dimensions. So to get 3-dimensional cells, we need to put them into a matrix. So we do electrospinning, which is membrane to support the cell's growth. We need to grow them in something. So there's this chip, organ on a chip, which is a microfluidics system where we can grow the cells, we can apply liquids with media to help the bacteria grow, to help the cells grow. And obviously then we want the bacteria to add the bacteria to this as well. This is a small picture of the system up and running. This has been done by loads and loads of work with Dr. Ruth McKay and her research. She spent a lot of time on the engineering aspect of this. So if you want to know more about the engineering, listen to her session in the SenGem session this afternoon. But I'm going to go and talk about bacteria now and trying to add the bacteria to this vagina on a chip system that is just being finalized and tested at the moment. So we've got this vagina on a chip. We've got epithelial cells growing. So these are the tissue cells that line your vagina. We've got the membrane supporting this. And hopefully, if it's all working well, you know, you have the mucus layer, which protects. Ronan mentioned about you having this protective layer on your skin. And what I want to do is I want to create a biofilm of lactobacillus growing on top of these tissue culture cells. Hopefully that will represent what's going on in the vagina normally. And then I want to try and challenge it with Gardnerella. So this pathogen we think causes bacterial vaginosis, but we're not sure. And we found that there might be different species of Gardnerella. So some may just happily live in your vagina, but others may be the ones that cause bacterial vaginosis. And what I want to do is add different Gardnerella strains and try and find out what is the reason for the cause of bacterial vaginosis. And as I mentioned earlier, there are over 400 different species of bacteria there. I would like to add a few of the extra bacteria that are quite often associated with bacterial vaginosis, and then try and make up this sort of simplified microbiome in the vagina. So for this, we've got to do a lot of optimization, and the research in my lab has mainly been getting the optimized conditions to be able to grow these bacteria at the moment. We know how to grow them normally, but we want to grow them under the conditions that the vaginal cells can grow. So I've cultured them, I've got them growing. As you can see here, these are growth curves showing that these bacteria are now happily growing under conditions that vaginal cells will grow. This conditioned media here that I've mentioned, this is where I've taken media that the vaginal cells have grown in and added it to the lactobacillus culture to see if it's any toxic effects, so whether the vaginal cells will kill them. There isn't, it's all looking good, so that's great. I've done the same where I've looked at Gardnerella and done exactly the same growth. With time, I can't really show you this data, but they're all happy as well, and I've optimized their conditions. And we've also had a look to see whether if we grow bacteria and take all the media and the things that the bacteria spew out, whether they impact on the growth of the vaginal cells. And at low concentrations, which we'd sort of hopefully expect to be what they're growing under, it all seems okay. However, we talked about biofilms protective. If we grow Gardnerella and lactobacillus together in a liquid and there's no biofilms involved, Gardnerella completely outcompetes lactobacillus. So lactobacillus needs this three-dimensional biofilm growing on epithelial cells, hopefully to be able to survive and protect against this invasion by Gardnerella. So the final things is the next steps we're going to be doing is growing these bacteria on top of vagina epithelial cells, compare how the different Gardnerella strains have different abilities to outcompete the lactobacillus and how do they cause an infection, and try and understand the genetics and establish the links of Gardnerella to find out specifically how it causes an infection and how it causes bacterial vaginosis. And finally, I just want to highlight, this is not me on my own. There are a lot of academics in Brunel working on this organ on a chip research. Dr. Ruth McKay and Dr. Silva are leading on this, but all these other academics are involved. And at the bottom, you can see we have a load of PhD students, engineering, biology, sort of looking at cancer biology, cell biology, microbiologists, all coming together to try and get this organ on a chip working. And in Dr. McKay's talk, she'll talk about the other organs on the chips that we're creating, but today I was just focusing on the vagina. So thank you very much. If you have any questions, do put them in the chat, and I'll hand over now to Professor Arturo Sala, who's going to be talking a bit about cancer and his research in that area. So thank you very much.
All right, so I'm going to give you a little, short talk about things that are going on in my laboratory. And although I do study cancer, it's actually cancer is a major interest, has been a major interest in my lab for many years. More recently, I've been involved in studying genetic diseases, in particular a rare disease called Friedrich Ataxia. And this is also because of historical reasons, and we have a very strong group. We were used to have one of the most prominent investigators in Friedrich Ataxia in our department, Mark Pook, who recently retired, but there is still a group, a very strong group led by Sarah Giovanni, who also studied Friedrich Ataxia. So I found this disease very fascinating. So, and I hope to illustrate why this disease is interesting. So as I was alluding before, I study cancer mainly, and in particular a couple of cancers that are rare but still have quite a big impact in terms of overall impact in cancer. Neuroblastoma, because it's a childhood disease, and adenoid cystic carcinoma, which is a head and neck cancer, and Friedrich Ataxia, as I was saying before. So let's start with Friedrich Ataxia. Friedrich Ataxia is a genetic disease caused by deficiency of an essential protein called frataxin. This protein, it is actually produced in patients, but the problem is that they don't make enough of it. So there is less frataxin in these patients. And the disease is incurable, and life expectancy is about 40 or 50 years of age. So some time ago, we developed a strategy based on the engineering of a therapeutic version of the frataxin protein that can be delivered by hematopoietic stem cells. And the protein has been engineered in a way to be at the same time secreted by stem cells, and also has the ability to penetrate tissues. So it's been quite dramatically modified in a way that we hope will be able to reach the target tissues that are critical in Friedrich Ataxia, i.e. the central nervous system and the heart. So this approach has been developed in collaboration with UCL and Great Ormond Street Hospital teams, and is based on the production of this modified frataxin by a lentiviral vector that is a clinical version of a lentiviral vector that is used to infect stem cells from patients, and therefore these stem cells are then reinfused in the patient, and where they hopefully will start to produce the therapeutic protein by differentiated hematopoietic cells in the different target tissues. Now, we are now at the stage of preclinical validation of the system. So the system works in mice, in a mouse model of Friedrich Ataxia. We are validating these experiments in further assays, but we hope to, in a not too long time, to be able to translate this strategy in patients. So we are starting collaboration with clinicians in UCL to adopt this new strategy. Now, briefly on neuroblastoma. So neuroblastoma is a very aggressive childhood cancer originating from the peripheral nervous system. As Felicity was alluding before in her talk, it is a rare cancer, but accounts for about 15% of overall childhood cancer mortality. So it is a serious problem in pediatric oncology. Standard treatments are quite aggressive cycles of chemotherapy, radiotherapy. So there are important side effects, taking into account that children and infants are treated with this very toxic treatments. So we wonder whether we could develop new strategies based on less toxic treatments. There is a little problem, though, that developing a new drug from scratch takes a long time. So therefore, we thought that maybe we could adopt a different strategy. So this figures just to illustrate the different stages of clinical trials. So we go from the preclinical stages in animals to safety studies in men, and this can take years or even decades to to become a reality in patients. So an alternative strategy is drug repurposing. So drug repurposing can drastically reduce the time required to bring therapeutic compounds to patients. And clearly there is no need to conduct safety studies because drugs are already existing. And so some time ago, we focused our attention on antipsychotic drugs because epidemiological studies suggested that patients treated with certain drugs, for example, Prozac, have a reduced cancer risk. So we investigated if Prozac could have anti-neuroblastoma activity. And another good reason to focus our attention on Prozac is that it is already used in children with depression and ADHD, and therefore it potentially could be quickly repurposed in the oncological pediatric setting. So to cut a long story short, we have used Prozac in different neuroblastoma settings, in vitro and in vivo. And we demonstrated in a recent study that Prozac reduces neuroblastoma growth, both primary growth and metastatic growth in these mouse models. And this is just a figure taken from this paper illustrating the effects of treating mice which have been transplanted experimentally with human neuroblastoma cells. And you can see here sections of the livers that are invaded in the control mice by different metastases, whereas mice that have been dosed with Prozac show a dramatic reduction of metastases in the liver. And yeah, that was the paper. And this study has attracted some, interest from news outlets, both nationally and internationally. So I will finish here with our future goals, which are to, continue the preclinical validation of drugs for neuroblastoma and other cancers. I had no time today of talking about our project on adenoid cystic carcinoma. So we want to finish the mouse trials for Friedrich Ataxia in order to, go into clinical trials in humans. And we are collaborating in this endeavor with, especially with UCL and the UCL Institute of Child Health. And yeah, I will finish here with the funders and collaborators in UCL, in Sweden, Italy, and Brazil. And again, I'm happy to take questions at the end with the other colleagues.
Good morning, everyone. This is Dr. Luigi Maggiotta Casaluce. I'm very happy to be here today to share with you our latest research on a fascinating but also concerning link between inflammation and depression and its relevance for the efficacy and safety of antidepressant drugs. Before starting to talk about the details, I would like to share with you two considerations. First of all, as Felicity mentioned earlier today, inflammation and perturbations of the immune systems are underlying features of many diseases. And at the same time, in the last few years, we started to realize that also a very high numbers of human pharmaceuticals can interact unintentionally with the immune system in a subtle way. So the aim of my laboratory, a big research interest we have, is to understand the complex relationship between drugs, inflammation, disease, and immunity, and to predict the implications of these relationships for both drug safety and drug efficacy. In the last few years, I started to be particularly interested in one specific type of condition that is linked to overall behavioral disorders, in particular depression and anxiety. If we look at the data published by the World Health Organization, the numbers are quite scary. This is a truly a global health challenge nowadays, and both depression and anxiety affects hundreds of millions of people all around the world. And there is an urgent need to develop novel preventive or even treatment strategies to tackle this issue. And one aspect that triggered my interest lately is that in the last few years, a growing body of evidence has highlighted the link between inflammation and depression. Now, we don't know whether the inflammation is a cause or a consequence of the disease at this stage. However, there are some interesting observations going on at clinical level. For example, we know that patients with major depressive disorder often display elevated inflammatory markers in the blood. Children that are 9 years old display elevated inflammatory markers and much higher probability to develop psychosis by the time they are 18 years old. And quite interestingly, certain type of immune cells and inflammatory markers in the peripheral blood can predict whether some patients are responsive or not to antidepressants. Now, how that is possible, we don't know, because the interaction between the peripheral immunity and the brain in a condition of health, is not, it shouldn't be as tight as this evidence may suggest, but it may be. Now, if we start putting the drugs into the game, we know that antidepressants definitely act on the disease because they are very effective sometimes to treat major depressive disorder. But we will show in the next few slides that they also act on both inflammation and immune-related pathways. From a public health perspective, the importance of establishing a relationship between inflammation and depression is due to the fact that many lifestyle factors every day trigger in our body what we call low-grade inflammation. It's a sustained low-level inflammation that is activated in our body. And these factors include pollution, chemicals in the environments, food, our diet, obesity, and even being overweight. These are all conditions that trigger inflammatory pathways in our body. Therefore, if we establish a causal relationship between inflammation and depression, this may open completely new avenues in terms of both preventive and therapeutic strategies. Now, I would like you to consider that up to 50% of patients who start using antidepressants do not respond to the first line of treatment. This is a very high number and highlights how we really need to identify novel strategies to tackle this condition. So what we do in our lab, we use evidence and methods at 3 levels. We use clinical data, we use computational biology approaches, mainly focused on network biology analysis, and then we use the Zebrafish model. And we're particularly interested at the moment in 3 categories of drugs, 3 classes of drugs: antidepressants, anti-inflammatory drugs like ibuprofen, and then steroidal anti-inflammatory drugs like some of the one that some of us use to treat, for example, asthma, like beclomethasone. And why the zebrafish model? You may be surprised, but the zebrafish has a lot of similarities with humans, and it has been used in biomedical science for a long time, and more than 10 drugs that are now have reached the clinics have been discovered using zebrafish screenings. But the zebrafish has 2 main features that are very interesting for us for this specific research. First of all, the larval stage is completely transparent. Therefore, we can use microscopy to visualize a lot of biological functions going on in real time in the animal. At the same time, using genetic engineering approach, we can label specific cell types. For example, in these images, you can see 2 types of immune cells, neutrophils and macrophages, and we can follow immune dynamics in real time in living intact organisms once, for example, we give the drug. At the same time, fish exhibit a very complex behavior, and we have a series of methods to generate a multidimensional profiling of fish behavior, which can be to some extent translated to human behavior. So these 2 features together make this model particularly attractive to us. So in the next few slides, I'm gonna quickly show you and discuss some of the results obtained in my lab. For example, one of the most exciting things we've done recently is we managed to develop a novel zebrafish model of inflammation-induced behavioral disorder. So what happens, taking the fish and exposing it to a pro-inflammatory compound that causes specific inflammation in the intestine, we can see that this fish also display a disruption of the behavior. In the graph on your left, you can see that there is a fish exposed to this compound display an increased infiltration of specific immune cells in the intestine, which we use it as a marker of inflammation. And the same fish then display an altered behavior. Behavior is quite complex to analyze because one specific behavior may not tell you too much, but as exactly for humans, when you take all behaviors together, then you may get a much better picture. So these graphs, it seems particularly complex, but in reality, it's quite simple. Each column of this graph is one fish, and each row represents a specific behavioral endpoint, a behavioral parameter, or an inflammatory parameter. So we use complex statistics to try to identify patterns in the data. So the columns indicated with the C are control animals. The columns indicated with the T are treated animals with an inflammation going on. So you can immediately appreciate, looking at the colors of these clusters, how there are some clusters dominated by control that display in a certain way, dominated by green color, but then inflamed animals start to behave very differently. So you can see these clusters dominated by treated animals that are clearly different from the other ones. So these provide us an in vivo model that can be used to test drugs that could potentially rescue this abnormal behavior. And the most important thing from a triage perspective is that we can do all this research using a life stage of the animal that is not considered as an animal, is not protected according to the law. So it's technically a non-animal testing. Working at computational level and data mining some large data sets generated by some colleagues in New York, what we identified, what we discovered is that many antidepressants display significant ability to interact with the immune system. And here there is a table showing you like a ranking of the different activities. You can see that some drugs are particularly active. This is the number of predicted immune interactions for clomipramine with 123 interactions. Other drugs are less active. And you can appreciate this difference between the drugs with the same antidepressants, even in this type of visualization here. Each column here represents one drug. Each row represents an interaction with a specific immune state. And we have 293 rows in different tissues and organs. And as you can appreciate, even visually here, some drugs have a lot of color going on, a lot of activity, whether for other drugs, there are a lot of black or non-active responses. So these indicates that different antidepressants have different ability to modulate the immune system. But then what we've done in collaboration with some colleagues at University of Liverpool, we specifically focused on the ability of antidepressants to trigger anti-inflammatory pathways. And similarly to before, we used complex bioinformatic approaches, and we ranked a group of 20 different antidepressants according to their ability to modulate immune and inflammatory pathways. And you can see as a snapshot in this figure here, we can say specifically with our analysis which biological pathways can be activated by the drug. So we now believe that these differences in the group of 20 antidepressants and these computational approaches can be validated using our in vivo model. But most importantly, we believe that our workflow may be able to inform preclinical and clinical decision-making in the short term, and establishing the causal relationship between inflammation and behavior may help us one day to use simply computational-driven approaches to identify the right drug for the right patient at the right time, at the right dose. And this is our long-term vision. So I think I would like to say thanks to my team and to my key collaborators and funders, both at Brunel and elsewhere, and I'll be happy to take any of your questions. So I think it's all for me today, and I will leave the floor to Raha and Felicity, who will talk to you about cardiovascular diseases.
So good morning, ladies and gentlemen. I'm very happy and privileged to be here and talk to you about our research on cardiovascular diseases. For those of you who don't know me, I'm Dr. Raha Pazoki, and I'm lecturer in biomedical sciences. So my area of research is focused on a risk of cardiovascular diseases, which is an important global burden. And cardiovascular disease kills 17 million people each year in the world. And if you compare this with around 2 million deaths from COVID in 2020, you can get a feel of the problem we're dealing with regarding cardiovascular disease every year. So cardiovascular diseases are usually preceded by various metabolic conditions, such as increased accumulation of fat in the body. For example, in adipose tissue, in liver, and in muscle cells. And these changes also cause various problems such as obesity, non-alcoholic fatty liver disease, and diabetes. So the other issue that we have with these metabolic changes is that they also increase vascular resistance, which is an underlying factor for increased blood pressure, basically. So cardiovascular diseases have multiple risk factors, as many of you know. Some of them are modifiable, such as sedentary lifestyle behavior, smoking, dietary habits, for example, alcohol consumption, salt intake, and unhealthy eating. And we also consider hypertension and diabetes as modifiable risk factors for cardiovascular diseases. And on the other hand, there are non-modifiable risk factors. These include age, sex, and genetic factors. And what we are doing in my group basically is to try and understand how these different factors play a role in cardiovascular diseases and what mechanisms are involved. So we are also interested in finding out the dynamics and interactions between these different factors in relation to cardiovascular diseases. So in our research, we use larger scale databases such as UK Biobank database, which includes basically over 500,000 participants from across the UK. And we put together several modifiable risk factors of cardiovascular diseases in a previous research, and created a lifestyle score. We used the scoring system based on the recommendations of the American Heart Association, and we found that poor lifestyle factors in combination increased risk of cardiovascular disease, myocardial infarction, and stroke. We also put together several hypertension genes and created a blood pressure genetic risk score. And we showed that this genetic risk score is linked to an increase in cardiovascular disease risk, myocardial infarction, and stroke. So based on this part of the research, we observed that both genetic and lifestyle factors worked independently on risk of cardiovascular diseases. Further analysis on this research showed that within different layers of genetic risk, adherence to an optimal lifestyle decreases susceptibility to hypertension, myocardial infarction, as well as a stroke. So as you can see in this graph, with, for instance, a person with high genetic risk score and poor lifestyle is in much higher risk of developing cardiovascular diseases compared to a person carrying same genetic risk, but they follow a more optimal lifestyle. So we then investigated the genetic factors involving dietary behavior, such as salt intake, for instance, and we did this by looking into genes that are linked to urinary sodium excretion. And here we identified 51 genes involved in urinary sodium excretion, and we found that these genes were overly expressed in brain regions, adipose tissue, and arteries. And we also found that these same genes were abundant in biological pathways that were involved in human behavior, as well as weight loss and body temperature. And our further causal inference analysis showed that those sodium excretions genes that were abundant in behavioral response were causal to hypertension and cardiovascular diseases. So we then looked into various resources and existing databases, and we found that the sodium excretion genes were linked additionally to metabolic conditions such as obesity, diabetes, hyperlipidemia, and accumulation of fat in various parts of the body. In an independent project that was linked to this, we also looked into the effect of genetic factors on liver function. And we did this by looking at variation in liver enzyme levels in relation to genes across the human genome. And with this project, we identified over 500 genes that were linked to liver function. And we showed that liver function genes were linked to similar factors as sodium genes, and these were accumulation of fat in the body, as well as accumulation of fat in liver, and diabetes, and hyperlipidemia. And we additionally found that these liver enzymes were abundant in a biological pathway called FXR pathway. And this pathway is known to be involved in metabolism of bilirubin, glucose, and lipid. And we know that changes in lipid and glucose are basically the main metabolic changes that we see occur prior to cardiovascular diseases. And our further causal inference analysis basically showed that liver function was causally linked to cardiovascular diseases. And the importance of our research here is in the fact that we link lifestyle factors to genes and to biological pathways. And finally, we show the causality of biological mechanisms to cardiovascular diseases. And this will shed light into further research and highlights specific targets to focus on in order to prevent cardiovascular diseases. And the paper of this recent work is accepted for publication in Nature Communication and will be release next Monday, basically. And here with my last slide, I'd like to acknowledge the Medical Research Council and as the funder of the project. And I'd like to thank the collaborators of the project in Europe and US, including the Million Veteran Program, the Rotterdam Study, Lifeline study, as well as the Northern Finland Bears cohort. And I'd like to thank you for listening, and I'd be delighted if you put your questions in the chat box, and I will be addressing those during the panel meeting. So I now hand over to Felicity for her talk on resolution of inflammation.
Right, because I'm quite conscious of time and I want to leave at least a few minutes if possible to have questions at the end, I'll make my presentation slightly shorter, just because some of it will be covered in tonight's open lecture. But I wanted to really get across some of the work that we do in the lab, in my lab anyway, and the research area that we work on. So we're very much interested in inflammation, and I should apologise, you will see some of these slides tonight as well. And so I'm sure a lot of you have seen this slide on the left, the picture on the left, the schematic showing the 5 cardinal signs of inflammation, those being heat, redness, swelling, pain, and in some cases, loss of function. So under normal events, inflammation and what we call the resolution of inflammation, so in other words, restoring to homeostasis, is a very positive thing. It's a good thing, and the body is very well equipped at doing that. So I put some pictures on the side here on the right that show different types of acute inflammation, from sort of skin inflammation to ingrowing toenails to effect of a wound on the eye, where you can see the pus there, the debris, the cellular debris that's there. And then also that's a picture of my hand where I had a little bit of glass in it, because which you can see the sort of inflammation that's there, and then over a few days later that inflammation has resolved itself. Now typically that's not necessarily the classic analogy that we do, but we normally say when we talk about acute inflammation, we talk about things like a bee sting, because quite a few of us have unfortunately had that sort of effect, and you get that classical sign, you get the swelling, the pain, that warmth that's there. And the redness that's there, not typically loss of function, but you do get certainly 4 of the 5 cardinal signs. And that normally over time then resolves under normal circumstances. So if you look at the graph at the bottom, it shows that you get, under normal situations, you get this onset phase followed by a nice, what we call resolution phase. So in other words, restoring homeostasis. The problem comes when we've got chronic inflammation, so we get this excessive inflammatory response, where there's a huge sort of upregulation of different inflammatory mediators, and that creates a non-resolving situation that occurs in a lot of chronic inflammatory situations. And my lab's very much interested in being able to manipulate that profile, if you like. So it's shown here in the sort of blue dotted line. So we use a pharmacological strategy, so we apply drugs to be able to help the body resolve that inflammation. And so there's a number of different chronic inflammatory disorders. We've mentioned some already within the context of the centre. Those that are here in red are more of the diseases that we're particularly interested in or have projects on at the moment within my lab. But we're looking at them in the context of resolution of inflammation. Yes. So really, there's a need for resolution pharmacology when it comes to sort of drug targeting and the approach to different chronic inflammatory diseases. And so my lab looks at the sort of understanding the physiology, the pathophysiology, the mechanisms and pathways associated with some of those diseases that I mentioned, in order for us to fine-tune better therapeutic targets, and in some cases novel drug approaches, in order to get better drugs to treat patients. We use a number of different approaches to do this. I'm not really going to go through them here right now, but one of the drugs that we've been looking at, which is very much involved in inflammation, is this annexin-1 molecule. It's a protein, it's stored very much in neutrophils, for example, and as Steve said, I'm a big fan of neutrophils. I'll talk about those more tonight. And it interacts on what we know is a receptor that's very much at the forefront of resolution biology, and that's the 4-mile-peptide receptor 2, or FPR2, as it's known, and annexin mediates its responses there. And we've shown very nicely using a number of different assays, from sort of in vitro approaches to in vivo rodent models of disease, as well as clinical samples, that annexin is able to modulate the what we call the phenotype of the cell, of neutrophils and of platelets, modulate those phenotypes so that in the context of the platelet, it presents or changes it such that it becomes more sort of attractive to eat, to ingest by the neutrophil. And so what we're interested in right now is really understanding how we can change the phenotype of different cells in the situations that they're presented in. And that goes from anywhere from the situation for COVID at the moment, for long COVID, as well as obviously other diseases such as even aging. Luigi had mentioned about this sort of low-grade inflammation that you have that in aging. You know, it's just part of our immune system. It's another comorbidity that's associated with the risk factors for certain diseases. So we're interested in that area. We look at everything from the microcirculation point of view. We're interested in cellular trafficking, the idea of neutrophils, in particular neutrophils, rolling along the endothelium. You can see here the picture at the top left shows a nice schematic of cells rolling along the endothelium. They adhere and then they migrate into the tissue. Each of those are mediated by different components, and we study that at the microcirculatory level. And by that I mean not just the circulation, not the arteries and veins, but we're going into arterioles and venules and capillaries, so the smallest vessels within the body. Because that's where it starts. So that's where the inflammatory components start. So before we get to the large vessels, before we get to those thrombotic states that occur, it starts in that microcirculation. So we use sophisticated imaging techniques in order to actually visualize in real time, in anesthetized animals, cells moving around the body, and we're able to look at those in the context of disease. So there's a very much a tie between inflammation and thrombosis, and by that I mean that a lot of the mediators and pathways involved in inflammation are also involved in thrombosis, and so you get this sort of one begetting the other. So you get this sort of thromboinflammatory component that's affiliated with a number of different cardiovascular diseases. I've shown here a little bit on stroke. I'm not going to go through it so much. I'll talk about it a little bit tonight, but really it's just there to say stroke's a classic example of a thromboinflammatory situation where we have this, the issues, you know, certainly surrounding stroke is there's two options. There's either surgery to remove that thrombotic clot in an ischemic stroke, so when there's the blood flow is stopped within the brain, and always time is brain, it's to reintroduce that blood flow in order to get the oxygen back to the brain as quickly and as effectively as possible. So we can do thrombectomies to do that, or we can give drugs. The only drug available at the moment or license is tissue plasminogen activator, and only about 10% of patients will get it, and only 5% of those that get it might have some positive effect. So really we're at a loss of finding or getting good therapeutic targets, and that's what we're looking at in the context of my lab. And we do, I won't go through this just because of time, but we do a number of different assays. We look at the cellular components. We know that the resolution profiling certainly in stroke is dysregulated. It is in a number of chronic diseases. We can measure that in the blood by looking at mediators of resolution profiles. We can see that neutrophils are activated. They're producing these what we call extracellular traps or nets, neutrophil extracellular traps. It's DNA that they throw out literally like a fishing net. Originally, it's really done as a positive mechanism, which is to scoop up pathogens, to ingest them in order for clearance, but they also provide a scaffold on which other cells can stick, such as platelets. And we can modulate the ability of those neutrophils to produce nets and the type of net that they produce by the treatment with annexin-1. So we do a lot of different sort of translational aspects and models. We use different species in our research, metabolipidomics studies, imaging, etc. And really the idea is for now and certainly the future is to look at data integration, Arturo mentioned about repurposing or repositioning of drugs, so drugs that are on the market already for perhaps a different disease state, of having a look at those in the context for us, in the context of resolution of inflammation, and also looking at different targets that might be available, or therapeutic targets. And that's it really. So obviously the funders that support the work throughout the course of this are listed there, and obviously I must thank those. Oh, I'm very conscious of we've run over time, and so thank you for everyone that has stayed. What we might do is answer the questions offline, because there are other sessions that are running in after this, and so I know that some people perhaps want to go to those sessions as well. So really, unless there's anything really burning, I would say thank you to everybody that's presented today. You've heard the sort of great work that's going on within the centre. We welcome the opportunity for networking, for collaboration, for further presentations to discuss the work in more detail. And obviously it remains for me really to thank those behind the scenes that have helped put everything together for the presentation today. So we're very grateful to them as well. Thanks everybody.
Great.
Take care.
Thank you.
Bye.
Yep.